Okay, good morning everyone. My name is Adam Auton and I'd like to start by thanking Mohammed and his fellow organisers for giving me the opportunity to speak to you today. And thank you to all of you for turning up first thing on a Sunday morning to hear a little bit about something that I've been working on just recently, beginning to look at recombination rate variation in the canine genome, which I hope to show to you is actually a relatively interesting topic, especially for people interested in recombination, which I hope people in this room are. However, I thought I'd start by just showing what recombination looks like in the human genome. It should come as no surprise to people in this room that recombination in the human genome is pretty much dominated by recombination hotspots. So here I am showing just some recombination rate estimates from the HapMap project that were obtained using statistical methods. And above each chromosome, I've got a little mark where we've called a recombination hotspot. And what this shows is that recombination hotspots are pretty much a ubiquitous feature of the genome. We call, I think, 30,000 of them, and we have perhaps 60% power. So we're talking, there are perhaps 50 or 60,000 of these things throughout the genome. And there's nowhere in the genome where there's more than about 200 kb where we do not have a recombination hotspot. However, for a long time it was a bit of a mystery as to why these recombination hotspots appeared where they were. All that began to change in late 2009, early 2010 with the identification of PRDM9 as a hotspot associated gene. And in particular, there were two groups working in mice models that identified that variation in PRDM9 caused variation in hotspot usage, and another group that using bioinformatics models were able to identify PRDM9 as predicted to bind to a previously identified hotspot associated motif. Now, PRDM9 has turned out to be a relatively interesting gene. It's made up of three domains, two of which have relatively high conservation across species, and this last domain here, this zinc finger domain, which really appears to be where all the action is. It's this zinc finger domain that is responsible for binding to the DNA motif that I identified before. And if we look at variation within this domain, within the human population, we see really quite high diversity. This zinc finger domain is encoded by a tandem repeat that's about 84 base pairs in length, and mutations can occur in this domain, and you can have rearrangements and deletions and duplications as indicated by this figure here. And what you can see is exactly as I just said, that there's all sorts of variation, and the length of the domain can vary from, I think, whatever that is, 9 zinc fingers to, I think, 13 or so. However, there is a dominant allele, which is this one here, which corresponds for more than half of the allele, the allele frequency is greater than 50% for that particular allele. If we look in another species, we see yet more diversity. So this is showing the diversity in chimpanzee, and importantly that there is no sharing between the human and chimpanzee PRDM9, and in fact chimpanzee seems to show a greater diversity in this zinc finger domain than does human. So whereas this is variation assessed in 148 human sequences, this is variation assessed in 50 sequences, and we detect nearly as many PRDM9 variants. And in this case, there doesn't seem to be a dominant allele as there is in human. So this was quite interesting. This began to explain an observation that had been kicking around for a little while, namely that despite having very, very high sequence identity, humans and chimpanzees really did not seem to share recombination hotspots. So here on the left, I'm showing recombination rate estimates from human along chromosome 21, and on the right, I'm showing some recombination estimates from chimpanzee. And if I line these two things up, you can really see that the recombination hotspots do not seem to line up with each other. We can formalize that a little bit, so we can essentially plot the recombination rate for humans shown as a black line here around human hotspots, and the chimpanzee rate in red. And around human hotspots, we see absolutely no elevation in the chimpanzee recombination rate, indicative that these hotspots really aren't shared between the two species. And likewise, we can plot the recombination rate around chimpanzee hotspots, and we see humans just completely flatlined. So these species really aren't sharing recombination hotspots. So this is kind of interesting. So PRDM9 is showing great diversity, and that seems to be driving differences in recombination. It goes a bit further than that. PRDM9 actually seems to be under really quite strong selective pressures. So I just wanted to highlight a couple of quotes here from a couple of papers. So this is a paper by Oliver et al. That was published, I think, in 2009, which considered diversity in PRDM9, or rather variation in PRDM9, across a wide range of species. And they have this nice quote here, that concerted evolution is also apparent in PRDM9 zinc fingers for many mammals, including elephants, cats, common shrews, cattle, deer, bats, and rabbits. So the selection's been going on for a very, very long time across the mammalian tree. Likewise, in this paper by Simon Myers, that was one of the papers I highlighted earlier, they identified PRDM9 as the most diverged of all of the zinc finger genes between humans and chimpanzee. And they say such rapid evolution is acceptable. Comparing these residues among all 544 C2H2 containing zinc finger proteins, human and chimpanzee ortholog pairs, PRDM9 is by far the most diverged. So PRDM9 is really under quite strong selective pressures. So it was something of a surprise that when people noted that PRDM9 is completely broken in dogs. So here I'm showing an alignment of the amino acids between humans and dog, and there are multiple disruptive mutations here. There's a premature stop codon up here and at least 4 frame shift mutations. And this is data that was presented by Axelson et al. In a genome research paper published last year. So that's very interesting. This is unique in the mammalian tree that PRDM9 is broken, and despite the selective pressures in the other species, somehow dogs have managed to survive with a broken gene. So we thought it'd be very interesting to look at recombination in dogs. Now, I thought I'd better start by showing that there is some prior art in this area. So specifically, there's this paper by Wong et al., which was published a couple of years ago, which was a pedigree-based map, so essentially an experimental estimate of recombination rates. This is based on 3,000 microsatellite markers using roughly 450 meioses. Now, they saw a relatively typical recombination landscape at this broad scale. They estimated a map length at about 2,000 centimorgans with a typical recombination rate of about 1 centimorgan per megabase. And there were other things that we would typically expect to see in a mammalian genome, that telomeric regions tend to be more recombinogenic than regions closer to the centromere. So things looked relatively typical. There's another paper that was published by Axelson et al., which was published in Genome Research just this year. And this is an LD-based study, so similar to the HapMap-based estimates I showed on the first slide. In this case, they used a microarray that contained 160,000 SNPs, or roughly 1 SNP for every 13 kb, and they were looking in nearly 500 dogs in 30 separate breeds. Now, at the broad scale, they were able to show that they have reasonably good correlation with the Wong et al. Paper, demonstrating that these LD methods may work reasonably well in this species. However, there's reasons to be a little bit cautious about estimating recombination rates from patterns of genetic diversity in dogs. Specifically, dog breeds have very, very strong inbreeding patterns. So here I'm showing tracks of homorygosity in this Labrador here, and as you can see in red here, there are these very, very strong, very long regions where you observe no heterozygotes, which are observed purely through inbreeding. So that's going to really make your life a little bit difficult if you're trying to use patterns of linkage disequilibrium to estimate recombination rates. And in fact, if one looks at the decay of linkage disequilibrium by physical distance, you can see that the breed dogs have LD that decays very, very slowly, expanding out here to about 100 kilobases. And if we compare that to, say, patterns that we see in wild populations, such as wolves and dingoes, it really extends very, very long, whereas the wild populations have a much more rapid decay. So there are reasons to be a little bit concerned about working with breed dogs. However, there's another population that we could perhaps use to try and overcome some of these issues, specifically the village dogs here. And what do I mean by village dogs? Well, these are dogs that have undergone domestication but haven't undergone bottlenecks that are resulting from breed formation. So this is potentially a source of genetic data that doesn't suffer from some of the issues that you get from working with purebred breeds. So with that in mind, I approached a collaborator of mine at Cornell, Adam Boyko, who I knew had been working on this project known as the Village Dog Genetic Diversity Project, where he's essentially collected samples from all over the world. He's now up to about 1,500 dogs from 25 countries. I think it's worth pointing out that this project is a bit of a labour of love, almost literally, in that Adam's family is involved. Specifically, his brother and his brother's wife have been travelling the world collecting samples and actually visited Namibia to collect dog samples on their honeymoon. So, you know, they really, they care quite passionately about this project. So we decided to take some of these dogs and see if we can identify genetic diversity in them and use them for constructing a genetic map. So specifically, we identified 35 Eurasian village dogs, plus some dogs from other species, and with other breeds, and we've sequenced them using Illumina HiSeq to about 8 to 12x coverage with 100 base paired end reads. We've done the usual things. We've aligned these reads to the dog genome using BWA. We did SNP calling using GATK and genotype imputation using Beagle. And in total, we've identified about 13 million autosomal SNPs, including 38,000 missense and nonsense mutations. And we've also done SNP calling on the X chromosome, but I'm not going to talk about that today, but just saying that that data is available should people be interested in it. Now, if we go back and look at what homozygosity looks in village dogs, we see something that's much more reassuring, as opposed to the very, very long tracks of homozygosity that we're observing in breed dogs. In village dogs, you really just don't see these patterns. So things are looking a bit more healthy in terms of being able to identify heterozygotes throughout the genome. And likewise, if we look at the LD decay, whereas before we had this very slow decay in breed dogs, there's quite rapid decay in wild dogs, related species. In village dogs sit quite nicely in between. So there's evidence here of the domestication bottleneck, but they're not suffering from the same breed formation bottlenecks that were potentially causing issues before. So we took this data and then we decided we wanted to construct a genetic map, and for anyone who knows my research, it would be no surprise that we used LD-HAT to estimate recombination rates, which is a statistical package that can estimate recombination rates from population genetic samples. We experimented with various thresholds for choosing SNPs and which dogs we included. I just kind of want to say that, you know, we spent quite a while doing this, but we essentially found our results are largely robust to whichever parameters we used. And so what do we finally end up on? Well, we performed extensive filtering of SNPs to ensure we really only used the highest quality variants for looking at patterns of LD. And specifically, we removed SNPs with excess heterozygosity, anything in a putative CpG site, which I've underlined because that'll become important later. SNPs in homopolymers, and so we really filtered quite heavily. We started off with about 13 million SNPs. For the purposes of building a genetic map, we just used 3.5 million SNPs, which is approximately similar to the number of SNPs that were included in the Human HapMap project. So we think that our genetic map will have pretty much similar resolution to the maps obtained from the HapMap project. So what does this look like? Well, so here I'm showing a comparison of the genetic map on chromosome 2 between our fine scale map in blue and the experimental map estimated from Wong et al. And I think you can see here this looks really quite good at the broad scale. These things track each other really quite nicely. Now, this might just be one chromosome, it might just be a very, very nice example, so I should really show you the rest of the genome. Now, in the majority of the cases, I think you'll agree it looks pretty good. There are some chromosomes where things seem to diverge slightly, but the overall picture is that really we're recapitulating the genetic map at the broad scale really quite well. We can formalize that. We can look at the correlation at various scales, and at the fine scale it's perhaps not unsurprising that we don't have particularly high correlation, but by the time we get out to about 5 megabases or 10 megabases, our Pearson correlation between the two maps is about 0.9. So pretty good overall. And I also wanted to put in this slide here, even though it's only tangentially related. So I wanted to put this slide in because I didn't really know where else to put it. But this shows the recombination rate by the chromosome length for each of the chromosomes. So you have the short chromosome 38 here and the long chromosome 1 here. And this shows that between the two maps there's relatively good agreement, with the short chromosomes having really quite high recombination rates, and the long chromosomes having quite low recombination rates, which is quite typical of what you would see in other mammalian species, perhaps associated to the fact that one requires at least one recombination event in each chromosome arm in order to ensure proper disjunction. So, what about if we look at things at the fine scale? Well, things look pretty hotspotty. This looks pretty similar to what we saw in humans. There appear to be recombination hotspots all over the genome. But, you know, really, can we quantify this in any better way than just showing the plots here? So, you know, we can say, well, how hotspotty are these really? So here I'm plotting a region of chromosome 12, and if I overlay behind that the estimates for the synteny region in humans, which is on chromosome 6, you can see that these dog hotspots are really actually quite small compared to the human hotspots. In reality, they're about a third of the size compared to the human hotspots. So what about, can we quantify this in another way? Well, this is a nice plot that I think is really quite informative. So on the x-axis here I'm showing proportion of recombination between 0 and 1, and on the y-axis I'm showing proportion of sequence. Now if recombination was uniformly distributed throughout the genome, we would expect these lines to lie right on the diagonal here. However, in humans we see that actually things are really quite skewed away from the diagonal, and about, what is it, it's about 80 or 90% of recombination occurs in just 20% of sequence. However, if we look at dogs, and I'm plotting each chromosome separately here, we see that actually recombination is much more uniformly distributed throughout the genome. It doesn't quite reach the uniform distribution that one would expect if things were just occurring randomly, but you know, really is shifted towards the diagonal quite significantly, and now 80% of recombination is occurring in about 50 or 60% of the sequence. Now, can we do further comparisons between dogs and humans? Well, yes, we can. Humans and dogs, despite being diverged quite considerably, do have quite considerable regions of synteny. So here I'm just plotting, or rather, I'm taking a figure from this paper here that shows the degree of synteny between the human and dog genomes. And you can see that there are even whole chromosomes on the left here, such as chromosome 12 shares quite a large chunk with, I think that's chromosome 6 there. So we can use this information to try and do some broad-scale comparisons between humans and dogs in terms of recombination. And so here I'm showing a couple of plots. On the top here, I'm showing estimates of the recombination rate in the dog genome, and under that I'm showing the recombination rate in the human genome over the syntenic regions, and I've just inverted it for comparisons purposes. And you can see quite nicely in the dog genome here that you have higher recombination rates towards the telomeres in pretty much all of the chromosomes. But if we contrast the patterns in humans, there's really no correlation between these two species. So, you know, they're quite diverged, and perhaps we would expect that there would be no correlation over such long evolutionary timescales, but I think it's quite nice to confirm that fact here. Just to contrast it, here I'm showing a similar figure between humans and chimpanzees. So here are the human recombination rates on the top, and at the bottom I'm showing the chimpanzee recombination rates. Obviously these two species are much less diverged, and the correlation between these two species is really quite high. So by the time you get to the separations between these two species, evolutionary conservation of recombination is pretty much completely extinguished at the broad scale. We can begin also to drill down at the fine scale with this genetic map, so we'll look at recombination rate variation around various features of the genome. So here I decided to sort of look at recombination rate around repeats, as this is something that proved quite successful in looking at humans. And we do see variation in repeats. So around this repeat we see a quite marked elevation in recombination rate, whereas around this repeat, which is an L1 element, we see a quite strong repression of recombination. However, it's worth noting that for all the top scoring repeats, they're all very CpG rich. This is the top scoring repeat here, which is CGG, just repeated some arbitrary number of times. And the recombination rate here can be about 6 or 7 centimorgans per megabase. So, you know, nearly an order of magnitude above the background rate. The fact that all these CpGs come out as top of the list is something that's quite interesting. CpGs are, you know, fairly unusual in the genome, so we can begin to try and delve into what's going on here. I can look at CpGs in other contexts. I can say, well, so I have all of these recombination hotspots in the genome. Is there any other sequence motif within these hotspots that is driving recombination? And actually what I find is that if I look at all these motifs, it really is the CpG signal that is jumping out. So here I've done a comparison of hotspots, and for each hotspot I've identified a nearby cold spot that I've matched for GC content and SNP density, and then essentially look for motifs that differentiate those two sets of sequences, and it really is the CpGs that are jumping out. Another way of looking at that is to say, well, you know, is it really CpGs or is it GC content? So on the left here I can look at correlation with GC content as a function of scale, and I can see in humans as I increase my scale, my correlation with GC content just goes up and up and up. However, in dogs it kind of plateaus and in fact actually drops a little bit as the scale increases. However, in dogs, if I look at CpG content, I see the similar thing here where it appears that GC content is just essentially a proxy for CpG content. In dogs, however, I just get this relatively uniform correlation with CpG content regardless of scale. Now, CpGs are interesting. They're not randomly located throughout the genome. They're actually preferentially located near transcription start sites. So this is really quite an interesting figure here. If we look at recombination around transcription start sites, we see this massive peak in recombination in dogs. Now, for many years we've known that there's actually a small peak in recombination around transcription start sites in human. However, it's quite clear here that this is completely dwarfed by the pattern that you see in dogs. Dogs are really trying to localize their recombination around transcription start sites. I can ask the question, well, you know, what's going on here? Can I localize this any better? So I can try and look at recombination around the various sections of the gene. So here I'm showing recombination upstream of the gene, and I can see as I approach the gene I get a peak in recombination. That peak in recombination is essentially maintained through the first exon, but drops quite dramatically once one moves into the first intron, and in later sections of the gene, such as the middle exon, the last intron, the last exon, I essentially am at the background rate. So the recombination rate elevation is really localized towards the start of the gene and is maintained throughout the first exon. Now this figure I think is really quite fun. I wanted to highlight a quote from this paper that was really just published last few weeks ago by Brick et al., which says, so they looked at the binding sites of PRDM9 in mice, and if they look in knockouts, they say, surprisingly, hotspots are still observed in PRDM9 knockout mice, as in the wild type. As in the wild type, these hotspots are found at H3K4 trimethylation marks. However, in absence of PRDM9, most recombination is initiated at promoters and at other sites of PRDM9 independent H3K4 trimethylation. So in knockout mice, you perhaps see a very similar pattern to what you see in dogs, which are essentially also a knockout of PRDM9, perhaps suggesting that dogs have adopted the H3K4 trimethylation marks that are associated with gene transcription. So I want to change track slightly now and talk a little bit about some of the evolutionary aspects of recombination and what influence it has had on patterns of diversity in the genome. So in this paper of Auton et al. That I've mentioned a couple of times, they essentially concluded that the death of PRDM9 in canids has resulted in the stability of the recombination landscape. You knock out PRDM9, PRDM9 doesn't change so much, and it doesn't essentially set where your recombination hotspots are in the genome. They based this conclusion by looking at patterns of substitution associated with biased gene conversion. However, the outgroup that they were using to look at these patterns was actually PANDA, because this was the closest species with a sequenced genome. However, these two species are separated by nearly 50 million years of evolution, so it's perhaps a little bit concerning to use such a distant outgroup. So just as a way of background for people who don't know or have forgotten, what do I mean by biased gene conversion? Well, it is essentially explained by this schematic here, where you get a double-strand break in the vicinity of a SNP, and this double-strand break results in the loss of one of the alleles of the SNP, which is then replaced based on the copying from the other chromosome. However, this results in a mismatch in these SNPs, and the repair machinery essentially favors G and C alleles as opposed to the A and T alleles, resulting in drive against the A and T alleles. So we wanted to look at this in dogs. But before I move on, if one looks at these patterns in humans and chimpanzees, here's the sort of thing you see. So here I'm showing the skew in AT to GC mutations around human hotspots in humans and chimpanzees, and what you can see is there's this quite strong skew in humans here. So humans are really substituting GC bases in around recombination hotspots, but you see no corresponding patterns in chimpanzee because they don't share recombination hotspots. There's no elevation of recombination in these regions. Likewise, you can turn it around and look around chimpanzee hotspots, and you see an elevation here. It's not nearly as strong as one observes in humans, but likewise there is evidence that chimpanzees have biased gene conversion occurring within their recombination hotspots. So in order to answer this question in dogs, we decided that we wanted to sequence something a little bit closer than the panda, and for this reason we decided to sequence the red fox, which is separated by about 10 million years of evolution. Also has a broken PRDM9, which is true for all canids basically, and we've sequenced this to about 11x using Illumina sequencing, again with 100 base pair paired end reads. And because we don't have assembled genome, we've mapped all of these reads to the dog genome, and we've called SNPs at both sites that are called in dogs, and we've done SNP discovery in the fox as well. So what does this look like if we now consider biased gene conversion in between these two species? So in this figure I'm showing the pattern of biased gene conversion, so the rate of AT to GC mutations relative to the rate of GC to AT mutations in dog, and this is around dog hotspots and cold spots shown in blue, and you can see in dogs we're seeing an elevation of AT to GC mutations around dog hotspots. Likewise, if we look at the same substitution patterns in dog hotspots, but looking at substitution patterns in the fox, we also see an elevation of AT to GC mutations, suggesting that foxes also share the same recombination hotspots as dogs. So this completely validates the conclusion that Axelson et al. Had, that recombination in the dog genome has stabilized since the death of PRDM9. So that brings me to the end of my talk. I don't know how I've been done for time, but it'll be time for questions. So just to conclude, dogs can provide insight into recombination that occurs in the absence of PRDM9 in a similar way that you might want to study PRDM9 in knockout mice. The death of PRDM9 has resulted in what I think is really quite an unusual recombination landscape. At the broad scale, things look almost typical of mammalian species, but recombination appears to be more uniformly distributed. Recombination localization does appear to exist, although perhaps not in quite the same sense that we're used to in looking in humans and chimpanzees, in that they have fewer and weaker hotspots, and those hotspots are perhaps slightly wider than one might expect in the other species. Canine recombination is strongly enriched around CpG sites, and in particular that's led to an elevation around transcription start sites, perhaps implicating that dogs are performing recombination by co-opting H3K4 trimethylation sites associated with gene transcription. And finally, despite high mutability of CpGs, strong signals of biased gene conversion are visible around dog hotspots, which fully supports the Axelson et al. Conclusions based on less fine-scale data than we have had here. So with that, I would just like to thank my collaborators, particularly Adam Boyko at the University of Cornell, and his collaborators at the BGI have been responsible for much of the sequencing. And I'd like to take any questions.